BIOL366 class. In lieu of our usual taking you down here and showing you our instruments and having you do hands-on taking images, we've had to be forced to do this by video. So we're going to do the best we can. My name is Dr. Ingrid Niesman. I'm the director here at the EM facility, which is located at the catty corner of the EIS building, engineering, and North Life Sciences. It's hard to find, but at some point in time you should try and come down and find us. So I've been here about 2.5, almost 3 years. We've been doing this same lecture and week for the last couple of years, but unfortunately we've had some issues this year. But what we're going to do is show you both the SEM and the TEM and show you the images and show you how it works. So I came here from UCSD, and I came here because I really like to teach, and I really enjoy teaching the TEM class, which we offer in the fall, and the SEM class, which we offer in the spring. We limit our students to 10, so it's a very personal one-on-one experience, and you get to learn how to use these marvelous instruments for yourself. So the one thing, since we're all worried about viruses and everything, you've probably seen a lot of EM images along with all of the news reports. So the one thing I want to show you is why EM is so powerful. And to do that, we will use this one. One of the things our students had to figure out. So what we do in EM is we want to figure out what the term scale means. And that refers to sizes of entities and how we end up looking at entities. So here is a cell. Okay? We can see a cell in light microscopy pretty easily. They're big. They seem small, but they're big. Here represents a bacteria in relationship to a whole cell. So you can see it's significantly smaller. You can make out bacteria by light microscopy, but you cannot see the parts of the bacteria. You couldn't see the flagellas easily, you couldn't see surface features, so it's very hard to distinguish. So EM is very good for looking at bacteria. Now a virus, like this little guy here, which is what is in the news right now, is so small that you cannot see them by light microscopy. We can only see them by EM. So it's about 100th to 1,000th of a size of a bacteria. So probably 1,000,000th of the size of a whole cell. So those are the sizes that we're talking about. So I'm going to come back over here and I'm going to explain to you the difference between a TEM, which is transmission electron microscopy, and SEM, which is scanning electron microscopy. So there are two different modalities that use the same principle of an electron beam to image our samples. So a transmission electron microscope, our sample sits inside the column and the electrons come and are shot through a sample, very much like a light microscope. Whereas a scanning electron microscope uses very low energy electrons and they hit the surface of a sample and bounce back off, and we basically vacuum them up, and that way we can see the surface representation of a sample. So TEM is high energy electrons, SEM is low energy electrons. And the other part of this that's really important is because they are electrons, we have to have a very high vacuum in order to use our samples. Otherwise, the electrons, one will come down here and hit an air molecule and bounce right off, and we'll no longer have a column of electrons. So what we're going to look at today are some cells. These are HeLAs here. We have a COS7 cell that we're going to look at in just a second. But what you're looking at are the surface features. You can see the morphology of the cells, you can see the little hairs, you can see the processes, but you're not seeing inside. You're only seeing the outside. And then the other thing we're going to look at on the TEM is right here. It's called a T4 phage, which is a virus of a bacteria. And we like to use this one because it has all kinds of little structure. If we come over here and we look at the diagram, which is included in your packet that we are sending, your content packet, we have a little head, we have a neck, we have this long collar and tail, and then we have this whole process here which injects the DNA directly into the bacteria. So you'll be able to see all of these on the TEM. So your guide for today is going to be my TA, Spencer. He's an undergraduate who I trained to do both TEM and SEM last year, and I was able to have him be my assistant for SEM this spring. He is going to be your guide on the SEM. And David Collins, a JDP PhD student from Marina Kalixnaya's lab, who has been my GA and TA for both of those classes in the past. And he will be your guide for the TEM. So, now we're going to go, and you guys can go with Spencer, and he's going to be your guide.
Hi, I'm Spencer. Again, as Ingrid said, I work here as the TA for the SEM class, and I'm going to be showing you basically how it's used and showing the cells on it. So here we have the SEM. Right here we have the column where the beam of electrons comes from, and in here is the actual chamber. So typically when we were to load a sample, you'd have to vent it, and then this would slide open, you could load your samples. We have a couple different detectors on here. One is a secondary electron detector, which detects more of like surface morphology features, and the other one is a backscatter electron detector, which focuses more on a higher contrast between different element Z numbers. So that can be more useful in material science and whatnot. With biology, typically we just use the secondary electron detector, which we're going to be looking at today. So right here we have our COS7 cell, and we're already pretty well focused on here. So this is our typical layout. We can see inside the chamber right here. So right there we have a picture of our chamber and where everything is, and then over here on the left we have a nav cam so we can tell which sample we're on and navigate between the two. Up here on the top left is our secondary electron detector, and here is our backscatter electron detector. So right now we're just going to bring this one up so you can see it a little better. And we're actually going to focus in on this guy in particular. So here over on the edges here, we have the Philopoda. You can start to look a little more at them. And again, as Ingrid mentioned before, we're looking at purely surface features. So one of the things that gives us contrast is something called the edge effect. So with sharp edges, you're going to have a brighter area. This is what we'll be able to actually distinguish in this picture. So if we zoom out here, we can see more of the entire cell. But essentially, we're able to view a lot of things that are very small in this. But one of the problems is that if we have a biological sample, we can't just put it in because it's not electrically conductive. So most samples that are biological, we need to prepare. I'm not sure how much your class will be discussing that, but there are different processes, and the TEM process is actually much more thorough than the SEM. The SEM is fairly simple in comparison. So that's one of the reasons that I really like using it, because a lot of the times it's, at least for other samples that aren't biological, it's as easy as putting it on the stub and putting it in the SEM. So it's essentially one of the things we do to make it conductive is we have a platinum coater in the lab. Typically you will coat your biological or otherwise non-conductive samples with a metal such as platinum or gold or silver. And this is basically so that you can have a coating on it that's very, very thin. Ours is 6 nanometers, so it doesn't dramatically increase the size of your sample, so you still get to see these surface features on it. But this way you'll be able to actually get some signal. If you were to put this sample in without a coating, you would get a lot of charging, which is basically charge building up on the surface, and you just wouldn't get very good signal to noise. But with this coating, we're able to see these features, and again, with very little addition of material.
Hi, I'm David Collins. I'm a PhD candidate here at San Diego State, and this is a transmission electron microscope. We use this microscope to look at small particles, proteins, and within the side of cells. Electrons are generated at the top of the scope and they're focused into a beam which then passes through your sample. Your sample is loaded on a specimen rod which is inserted inside of the microscope in the center of the beam. That image that forms as the beam transmits through your sample is then focused and then magnified and viewed below. The image that's formed of your sample is collected on a camera or can be viewed in this fish bowl below. At low magnifications, viral particles are indistinguishable, but as we zoom in, we begin to see individual viruses. And then you can stop it.